Alright, I am going to talk about mitosis. So you guys have already learned about the cell cycle. You remember that it has three main stages: interphase, mitosis, and cytokinesis. So here's the big loop because it is a cycle, right? So we go all the way through interphase, and during interphase we grow a lot, we make copies of our DNA, we make copies of all of our organelles, and then we head into the actual mitotic phase or mitosis phase. And mitosis is where the cell actually goes through the process of splitting. Finally, it'll go into cytokinesis, and cytokinesis is the final stage where that one cell turns into two complete cells, and then each of those cells will go through their own cell cycle. So remember, interphase has the growth where we increase in size, we copy our genes or make copies of our DNA molecules, and then we grow and make copies of organelles. This is what the cell looks like during interphase. All of our DNA, if you notice, it's really stringy and kind of scribbly in there. And that's what chromatin looks like. If you remember when we did cell organelles, chromatin is in the nucleus. Chromatin is uncoiled, unthickened DNA, so it's really thin and stringy. And during interphase, this is what it looks like. So if we were to look at a cell in interphase, we would not be able to see anything in the nucleus, okay? It would just look like nothing. These guys over here are centrioles, okay? We would be able to see those because they're in every cell, but we would not be able to see our DNA. After we go through our interphase where we grow, copy our genes, and make copies of organelles, we will move into mitosis. So mitosis is a continuous process, but it has been divided into some stages, 4 main stages to be exact. You have prophase, which is what we start off at. So interphase goes right into prophase. And then prophase moves into metaphase, and then metaphase moves into anaphase, and anaphase moves into telophase, and telophase moves into cytokinesis, which moves into interphase again. It is a big cycle. So let's talk about the different phases. So the mitosis phase 1, so the very first phase of mitosis is called prophase. P-R-O. Pro actually means first. So prophase is the first phase. This is the first and the longest phase, and this is when that chromatin, if you remember what I was just talking about, all this squiggly stuff right here is going to thicken and become visible into these things called chromosomes. Okay, remember we talked about those too. Chromosomes are thickened DNA, so as the chromatin was thin DNA, once it spindles up and winds itself up, it's going to thicken itself up, and we are going to turn into chromosomes. Okay, each chromosome, okay, consists of sister chromatids joined at centromeres. So we have copies. So this is what our DNA looks like. We go through DNA replication, remember that's a part of interphase, and we end up with something called sister chromatids, which is 2 copies of the exact same chromosome. Okay, and now we go through mitosis, and it's going to actually pull these 2 apart. This one's going to end up in one of the cells. This one's going to end up in the other cell. So these are daughter chromosomes. If you'll notice, there's also this piece called a centromere. The centromere is where the 2 sister chromatids, so the 2 copies of the same chromosome, are joined together. And that's important because that's how the cell makes sure that each cell gets a single copy of every chromosome. So during prophase, we have these sister chromatids that are all ready to go. They're all joined at the centromere. Then also the nucleolus. So remember how we said there was the nucleus, and then inside was this area called a nucleolus, this even center of the nucleus. And that's where ribosomes were made. Well, the nucleolus actually disappears, so it kind of disintegrates, and the cell stops making ribosomes. The football-shaped structure called a spindle. The spindle is almost like spider webs, like little fingers reaching out from those centrioles. Remember, these are the centrioles. So they're like little fingers that reach out from the centrioles, and they start tugging on these chromosomes at the centromere to get them all lined up along the center. So this is what the picture of prophase looks like. You'll notice that we have our sister chromatids. So this is, let's say, chromosome 1, and this is the copy that was made of chromosome 1. They're attached at the centromere. Chromosome 2, copy of chromosome 2. chromosome 3, copy of chromosome 3, and so on and so forth. And you'll notice that this is the nucleus and it's starting to break down. Also notice that the centrioles are sending out these little spindle hairs, and those spindle hairs are going to attach to the centromeres and get them all lined up for metaphase. Metaphase is the next phase. It is a brief second phase, so while prophase was pretty long, metaphase is reasonably short. And this is where all the chromosomes are put into the center of the cell by those spindles. Okay, and they're going to line up along the metaphase plate and be completely in the middle. So here's the picture. Notice how our spindles or our little hairs have gotten really long and they attach. Okay, this guy attaches to this side of the centromere, this guy attaches to this side of the centromere, and that happens at every single chromosome. There will be a spindle from this side and a spindle from this side, and they're all lined up along the middle. And when they get the signal, those little spindles, just like a fishing wire, are going to go. And when they go, they're going to yank on the side of the chromosome that they have. So this is anaphase, the third stage where these sister chromatids are separated, and each chromatid is now considered a daughter chromatid. I don't know why they didn't use son chromatid, but that's what they use, is daughter chromatids. It just means it's the new chromosome for that new cell. The spindles also begin to shorten, pulling the daughter chromatids towards opposite ends, or the poles. So again, each of those centrioles are reeling in each of these chromatids, daughter chromatids, like a fisherman reels in a fish. So here we go. These little things are starting to shorten, and look, they pulled them apart at the centromere. So now you have a daughter chromatid going to this side of the cell, and that same copy of the daughter chromatid going to this side of the cell. And that's how the cell organizes and makes sure that all of the necessary DNA ends up in every single cell. So all of these guys are on this side, all of these guys are moving to this side. Also notice the cell is starting to get a little bit longer and starting to pinch a little bit in the middle. The next phase is telophase. This is the final stage where the chromatids are at the opposite ends of the cell, and two nuclei are formed around each of these new sets of chromosomes. The two daughter cells are formed when new double membrane forms, allowing cytoplasm to divide and separate by cytokinesis. So cytokinesis is not a part of telophase. It's the next phase in the cell cycle. But at this point, all telophase is just where these chromatids are at the opposite end, and there's now nuclei that surround them to protect those chromosomes. So notice this has elongated more and started to pinch more in the middle. The spindle's completely gone. And notice that the nucleus is beginning to reform. So this guy has a nucleus, and this guy has a nucleus. Also, you can start to see a faint line here, and that faint line is where the cytokinesis is actually going to happen. So remember that cytokinesis divides the cytoplasm, and basically a new membrane grows in between those two. Okay, and then it basically pinches off, and each cell has its own. Also notice that the chromosomes that you could see a second ago as very thick lines are now decondensed. They've kind of turned into that spindly, loose DNA again. And that's a good thing. That's what's supposed to happen. And as soon as we're done with cytokinesis, it starts all over again. We move back into interphase. So each of these two daughter cells will each move into their own cell cycle. So a quick flip through the whole cycle so you can see it all go together. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Once again, I want you to see it. So remember, this is a part of the cell cycle. So interphase, growing, developing, copying DNA, making copies of organelles. Then now we're in the mitotic phase, or mitosis. Okay. You've got all the chromosomes, they're in the middle, the spindles are beginning to go out, the nucleus is beginning to disappear. Metaphase, where they all line up along the center, and the spindles grab grab a half of each of the sister chromatids. And then anaphase, they pull them apart. They start reeling them in like fish, and these guys go to this side, and these guys go to this side, and the cell begins to pinch in the middle. We've got our nucleus that begins reforming around our new DNA. And the cell is pinching even more, and then cytokinesis, the cell membrane splits. okay, or the cytoplasm completely splits and we have two new cells. So we'll watch that one more time. Interphase, mitotic prophase, mitotic metaphase, mitotic anaphase, mitotic telophase, and cytokinesis. All right, I hope that made sense. Thank you.